right? I don't know why they did this. They have to rearrange it.
It has to be in order. Are they not following alphabetic order?
No,
Okay.
so probably I should create a folder.
So when it's my time to present, you're going to open my presentation, or what will happen?
Yeah, so. Just a minute.
Okay.
Get it. Glass your cup. I'll be right back. Maybe I can do this show.
What's happening?
So, this guy would have to present first, and then this one, and then this one. I just like that names are different.
So I know this one should be right here.
Yeah,
And one other person, I believe it was Rachel Kaiser. Hers is right
Yes.
there. And then I haven't seen this person, but I believe that it might be right here.
yeah, so if they are presenting,
Mhm.
how is it? Has it been already in order?
Um, have they been up here already?
Mhm.
Have they been up here already, or how would they present?
Yes,
How would they present?
in order? Yeah.
Okay, so all they have to do is come up and pull up their PowerPoint. So let's see, first is Rachel Kaiser.
Mhm.
So she just double click on her PowerPoint, that'll pop up.
Okay, so you just see your whatever, and then you present.
Mhm.
Oh, okay, that's fine.
Yeah.
Yep. Okay.
And I guess I can go ahead and leave that up. All right, let's know if you have any other questions.
Thank you.
I'm probably gonna wait maybe one more minute for the stragglers. Start in a minute or so. Okay, I think we'll get started. Welcome to the antimicrobial resistance session for speakers. And I'm Mark Sopsey. I'm a faculty member in the Department of Environmental Sciences and Engineering in the Building School of Public Health here at UNC and affiliate of the Water Institute. So it's my pleasure to welcome you to this session. We have 3 speakers. Each speaker is asked to speak for between 10 minutes. There'll be a couple minutes for questions, and then we will move on to the next speaker. And then at the end of the presentations, there will be a panel discussion with the speakers. Probably up here. Hopefully, there'll be, as you see, a microphone available back there. So if you have questions, then you can use the microphone. Our first presentation today is by Rachel Kaiser, who is from Tennessee Technological University, and her title is Antimicrobial Resistance and Environmental Health for Water and Stewardship Training.
All righty. Is everyone able to hear me? Okay, just making sure that works. Yeah, so good afternoon. As you mentioned, my name is Rachel Kaiser. I'm going to be presenting today on a chapter from my dissertation research, which is Antimicrobial Resistance and Environmental Health, a water stewardship framework for global and national action. This has been done in collaboration with the United Nations University Institute for Water, Environment, and Health, McMaster University, and again, my home institute, Tennessee Tech. So I just want to start us off with a little bit of background and why this is even relevant. So antimicrobial resistance, or AMR, does occur naturally, but overuse and misuse are accelerating this natural phenomenon, specifically use in human and animal being the primary contributors to this, with human consumption increasing 65% from 2000 to 2015, and the use within animals has tripled that, the use within humans. And this is a concern because these resistant genes, resistant bacteria, these antimicrobial compounds are then able to be introduced to the environment, specifically water resources, through treated and untreated wastewater, runoff, and pharmaceutical production and other industrial manufacturing. So with that, AMR has been detected throughout the water cycle in environmental waters, wastewater, and water supply. And this is a concern for public health because these water resources act as both a reservoir and also a pathway to humans through consumption, wash resources, and recreation. So what is being done to kind of manage and mitigate this? So globally in 2015, the World Health Organization established the Global Action Plan with 5 objectives, including improving awareness and understanding, strengthening surveillance and research, reducing the incidence of infection and also the need to use antimicrobials, as well as optimizing that use. And lastly, ensuring that there's sustainable investment in countering this resistance. And the goal of these 5 objectives are to be addressed through a One Health approach, considering human, animal, and environmental health sectors. And so this is kind of organized and regulated by this tripartite, and their job is basically to provide global guidance to nations so they can develop their own national action plans to combat AMR through stewardship. Oops. So our kind of purpose and goal was we wanted to understand what the priorities are within these stewardship plans and what are the gaps, kind of looking at them from all One Health sectors and also taking an in-depth focus on water resources. So we developed the One Health, One Water AMR stewardship framework. So this framework, we did a massive literature review and consolidated pre-existing frameworks and kind of modified this into our framework that includes One Health sectors, stewardship themes, stewardship pillars, and levers and sub-levers. So just to kind of bring that all together, this is what the framework looks like overall. So kind of breaking this down. So we have our One Health sectors and the themes that are associated with each of those sectors. And then we have our 4 stewardship pillars that have their associated levers here, the top line within each of those rows. And then we have our sub levers, which are the evaluation component of this framework. So these are then applied to each of the One Health sectors, themes, and also the stewardship pillars. And then if appropriate, there is a water specific complementary sub-lever to each of these questions. So with this, we would then evaluated national action plans based off of this criteria, ranging from either not included within the country's national action plan, all the way up to there's an established plan and their stewardship program. And then kind of going further into the water resources aspect, we considered water from the three main roles it plays in society as either a source, supply, or wastewater, and then the types of water within those categories were also applied to this evaluation, so we were able to really see where these priorities and gaps are aligned within these stewardship plans. And then these were all broken down into scorecards, so we were able to compare the results from the One Health sectors and themes and within national action plans themselves. So after we created all that, we decided let's apply it. So we selected 25 country national action plans as our case studies, and these they include all three World Health Income classifications as well as all 6 World Health regions. So now going into the results from our study. So first looking at the human sector of One Health, this sector is concerned with monitoring the development of and a dissemination of AMR that's associated with human health, as well as impacts of AMR to human health. So as you can see here off of our scorecard, so these are all broken down where we will have our sector here and then associated themes within that sector, and then the rest of the scorecard kind of showing those results. But we're able to see that this is prioritized within these national action plans, and then specifically the human IPC theme is the primary focus of all the national action plans we considered, regardless of region or income level. The next theme, the management of consumption in humans, is also highly prioritized. We did note that there were outlined actions more in middle income and high income countries than low income. And we kind of determined that this was because of limited implementation and regulation resources within these countries, but there is also ease of accessibility. So this is kind of highlighting one of those gaps here within this theme. But overall, for this sector and themes, there's universal implementation within AMR stewardship for human health. Then we broke it down by our animal One Health sector. So this one is concerned with monitoring the development of AMR within animals and how that is a potential exposure pathway to human health. So within this sector, looking at the theme of use of antimicrobials in animals. So after human IPC, this is the second most prioritized theme within these national action plans. However, this is focused on livestock and food animal production. So it's overlooking wildlife as well as companion animals within this theme. But overall, this is universally targeted. Looking at the next theme within this sector, food safety and security overall has received limited attention compared to the pre-existing themes that we've discussed. So kind of highlighting this as a potential gap within AMR stewardship. And then lastly, within this sector, the theme of use of antimicrobials in plants is highly overlooked and pretty much not even included in a lot of these national action plans. So this is a serious gap within AMR health and stewardship that we kind of highlighted through this evaluation. But overall, the animal One Health sector tends to be prioritized as well within AMR stewardship. So now diving into the environmental sector, this is concerned with the environment acting as an AMR pathway to human exposure. As you can see here, we have pretty much limited attention to this sector overall. And then kind of looking down into some of these themes, the environmental contamination theme considers AMR in air, soil, water, and the environment as a whole. There is limited inclusion of this within national action plans. And if it was included, it was pretty much related to wastewater resources. And if these regulations did exist, we kind of saw them in high income countries as well as middle income countries related to human and animal wastewater treatment. Kind of an exception to this as a low income country is India, where they had established regulations for pharmaceutical wastewater, but their limitation was implementation and enforcement, lacking those resources. So even though the stewardship action was defined, we're still seeing this gap of seeing it all the way through to being effective. And then also looking at the clean water and sanitation theme. This is concerned with access to clean water supply specifically that's used to reduce AMR infection as well as through wash measures in both human and animals. And again, this kind of ties back into protecting this water supply and human health aspect. So still kind of really overlooking the limited concentration on these environmental health problems. So overall, we saw that there was limited action universally for this sector as well as associated themes. Interestingly, though, there was greater concern in our LMIC countries compared to high-income countries as a result that there's limited access to WASH and a higher risk of AMR exposure here. So we're seeing that there's concern here, but there's an issue of resource availability and implementation. So diving further into water resources, as I mentioned, we looked at water resources within each of these national action plans and saw that their primary focus, again, is wastewater, if it was mentioned, limited focus on water supply or drinking water, and then environmental water is being significantly overlooked. If included, it was surface waters, but otherwise overlooking these other water resources such as groundwater here. And then wrapping up here, taking a step back, looking at our 4 stewardship pillars. So the first being mitigation. These are stewardship actions for regulation and management. These were prioritized in our low and middle income countries, but in relation to human IPC and consumption. The innovation pillar is concerned with research progress, AMR understanding, as well as data sharing networks. And due to resource availabilities and technologies, these tend to be prioritized in our high income and middle income countries. The prevention pillar, our stewardship actions kind of related to proactive actions, public outreach and education and training. These are pretty low resource intensive. So these tend to be prioritized in our low and middle income countries. And then lastly, surveillance is concerned with monitoring AMR and using that data to influence the actions of the other pillars. And we see that this is also prioritized in high income countries due to that resource and technological availability. But there is global support for surveillance that low-income countries are able to use as well. So just overall our findings, we see that the One Health lens is biased towards human and animal health. But within that, there are gaps for food safety and security, as well as antimicrobial use in plants. Within the environmental sector, which in itself is overlooked in stewardship, there is a limited water lens that's biased towards wash and wastewater management. But there's still limited action within those practices compared to human health aspects. We noted too that environmental waters are overlooked and that there are, within that, the current water related AMR guidance reflects siloed management. So we're looking at water resources as either a supply, wastewater, or source. So these need to be equitably treated as being potential AMR reservoirs and exposure pathways. So overall, One Health, One Water, and the stewardship pillars need to be holistically integrated, both globally and nationally, in order to protect and manage water resources effectively against AMR. I know that was a lot all at once, so I'm sorry. Any questions?
We probably have time for a question too, so.
Have 3 minutes. Okay.
Wescombe from American Society for Microbiology. Thanks for that presentation. Could you talk about the methodology a little bit more in terms of, I understood you did a literature search of all the NAPs, and how did you, it's like a read through many of or most of them, or do you have like a scanner to go through and pick out keywords to say, yes, this NAP has checks marks on the metrics?
Yeah, absolutely. Like I said, I kind of condensed that down within that. So you're asking more about the case studies we selected to apply the framework to, like how we chose those case studies. Yes. Okay. Yeah. So with that, I basically had a selection criteria. So if the National Action Plan was included within the World Health Organization's Library for National Action Plans, it was either available in English or translatable into English. It was currently active because a lot of countries have ones that have expired. So that was one of the criterias. And then also we streamlined it down since we had a water focus, if it had water related terminology within its national action plans. And that was just the manual screening on my part. So that's how we narrowed it down to those 25 countries within that, because I think there's over probably about 117 or 120 that have currently submitted or in the past participated in this program.
Other questions?
Hi, thanks so much for your talk. I'm Heather from UC Berkeley. I was really curious about the use of antibiotics in plant production. Do you have a sense of like what the scale of antibiotic use is in plant production? It seems like it's kind of not well estimated. But yeah, I don't know anything on that.
Yeah, so with this, like so there was a lot of literature review on my part. And so in the global guidance provided by those world organizations, they have addressed this, but there's really not a lot of data to state how much is really out there, but they do know kind of it's applied to plants similar to with livestock. So as a proactive measure to make sure you don't lose your crop and you can maximize the production of it as well. So with the issue with that is potential of runoff through precipitation in that aspect, but then also through exposure to human health. It can be consumed through consumption of that plant product. So plants are able to take any of these antimicrobial compounds that may be in the soil, they can absorb through their root system and integrate into their leaves through that method, as well as through application. So they don't really know kind of amounts yet because it's really overlooked, but we do know that it is an issue and a gap that needs to be addressed.
Okay, thank you, Ms. Kaiser. Go ahead. We're going to move on to our next speaker, Eugene Liu from Ohio State University. The title is Advanced Efficacy of 222 nanometer over 254 nanometer UV on antibiotic resistant bacteria inactivation, resistance genes damage, and inhibition of resistant gene transfer.
Hello, everyone. Thanks for the introduction. So I'm Yijing. I'm a PhD candidate from the Ohio State University. So instead of like focusing on the public health of the ARB, today I'm going to talk about how to treat the ARB with UV disinfection. So I will compare two like disinfection on the ARB with 222 excimer lamp and 254 low pressure mercury lamp. So like according to the World Health Organization, the antibiotic resistance has become a critical global public health concern of the century, and the overuse and misuse of antibiotics has promoted the evolution of bacteria and has selected for some resistant bacteria. And because of the resistance, the ARB exist in like various environments, including soil, lake, river, coastline, sewage, and even exist in like treatment systems such as wastewater treatment systems and like tap water. Like several studies has indicated that even after water treatment, the effluents of the treatments still detected a lot of ARB. And there are around 2 million infections and 23,000 deaths annually, like due to the ARB infections. And the ARB infections can like result in like infections, breaks, and like will have like make it a higher morbidity, mortality, and hospitalization. And the biggest concern of the antibiotic resistant bacteria, or the biggest challenge part to treat them, is that they can share their like resistant genes among different bacteria. So the UV disinfection is an advantage disinfection technology compared to traditional chlorine, because UV disinfection, there's no chemical like added in the system, and also like during the whole disinfection process, there is no byproducts produced. So like in like now like most like UV light sources are the low pressure mercury lamp, which has a 254 nanometer wavelengths. However, the mercury is not environmental friendly and also toxic and can cause cancer to human health. So like the study of our project is to explore another like UV wavelengths to like instead of the low pressure mercury lamp to see if it can apply it in the like real conditions. So here is a like literature review that we like referenced a lot that they did like they exposed the Bacillus 1A189 to different UV like ozone, chlorine, different like disinfectants. And like this Bacillus subspecies 1A189 is a multi-drug resistant bacteria. And in their finding, like at a typical UV dose that used in the waste treatments now, the 40 millijoule per square centimeter can obtain a 2 to 3 log reduction for extracellular gene damage and can obtain 1 to 2 log reduction for intracellular ARG damage. And for our study, we obtained similar ARG damage results on the 254 nanometers. And we also further explored the UV disinfection with 222 nanometers, and I will talk more in the later slides. So like talk about the mechanisms of how UV works. The UV light inactivates microorganisms as a result of damage to nucleic acids and proteins. So you can see in this finger that the blue line is like absorption by DNA, and you can see there's a peak absorbance at around 254 nanometers. But for protein, like they had a high like peak absorbance at 222 nanometers. Since in the like real conditions, you cannot treat the DNA like only, so we are thinking about to like explore the lower wavelengths on the like bacteria, because bacteria have proteins and DNA together. And for the DNA damage, it like damage the DNA damage was created by the creation of the cyclobutyne pyrimidine dimers. And for protein damage, it was caused by the damage to amino acids and peptides. So based on what I just talked about, we have the hypothesis that wavelengths below 230 nanometers will have more disinfection effects. So for our study, we still choose the Bacillus subspecies 1A189 as our target. And we expose the cells and or its extracted DNA to either 222 excimer lamp or 254 low pressure mercury lamp. Then we quantify the disinfection in 3 ways. The first one is to like quantify the UV disinfection of the inactivation of ARB. And we use a culture-based quantification method to like counting the CFU before and after the UV. And the second part is to quantify the gene damage after UV disinfection. And we use the continuous polymerase chain reaction, also known as qPCR, to like analyze, to like quantify the gene damage. And we also have 4 different sites of primer, which targeting different lengths of the gene, like on the gene region, to like analyze if the length of the amplicon will like have an effect on the gene damage. And as I like introduced, the gene transfer is a big concern. So we also quantify the disinfection with the inhibition of horizontal gene transfer. So we like combine the resistant genes with a recipient, the 1A1. The 1A1 recipient is a non-resistant bacteria, and we grow them on a selective growth medium, and the second growth medium has antibiotics. So if there's only 1A1 there, the 1A1 cannot grow because the antibiotics will kill them. However, if the genes, the resistant genes has been successfully transferred to the 1A1, then you will observe the growing of the bacteria on the plates. So it goes to the results parts. For the ARB activation, we noticed that as the UV doses goes up, the inactivation efficiency goes up. And compared between the two UV wavelengths, we found that the 222 nanometers shows greater disinfection efficiency. And for the antibiotic resistant gene damage, for the extract DNA directly, this is example of the gene damage results at 260 base pairs, the shortest amplicon base pairs, and we found similar disinfection results between these two wavelengths. And also we have analyzes the results under 4 different amplicons, and we didn't observe a significant difference among them. And for the intracellular DNA damage, that means the cells exposure to the UV, then we extract the DNA and quantify the DNA damage. And similarly for the 260 base pairs, we found that the 222 nanometers had a better DNA damage effect compared to 254 nanometers. And for the whole picture from the shortest amplicon to the longest amplicon, 222 nanometers showed a great damage for both for all the cases. And if you compare among the amplicons, you will notice that longer amplicon lengths higher DNA damage were observed. And this was because the longer amplicon will increase the detection probability during qPCR quantification. And for the horizontal gene transfer inhibition, and for both cases of extracellular DNA exposed to UV and the cells exposed to UV, we noticed that 222 nanometers showed great horizontal gene transfer inhibition compared to 254 nanometers. So from these results, we can conclude that 222 nanometers had an overall better performance on inactivation, intracellular gene damage, and horizontal gene transfer. However, during the experiment, we noticed that to have a same UV dose, we have to exposure the samples to 222 nanometers for a longer time. For example, if you want to obtain 40 millijoule per square centimeters, for low pressure, you only need to expose to 3 minutes. But for 222 nanometers, you have to expose to 12 minutes. So we have to consider about the energy consumption by these two wavelengths. So we calculate the electrical energy per order, and the electrical energy per order was related with the water volume we used during our UV experiment, and also related with the water factor and the wall plug efficiency of the two wavelengths. And we got these results. And you can here we show 3 cases: the ARB inactivation, extracellular horizontal gene transfer, and intracellular horizontal gene transfer. In all the cases, even though the 222 nanometer has a better performance, the 222 nanometers need higher energy to get the same disinfection effects. So based on results, although this is only a bench study in the lab, it still can indicate something to apply the UV in the real fields. So the combined DNA and protein damage by the 222 nanometers could explain the better performance on ARB inactivation, intracellular gene damage, and horizontal gene transfer. And when apply the UV systems to the fields or to the real conditions, we have to balance the disinfection effects and the energy consumption. We have to think about if it is worthy to like spend a lot of energy or like money to have a like better disinfection results. And because this is a bench study, is a culture that we used is at a very high like concentration dose. But if you think about the natural water or the wastewater, there are a lot of like particles, like other waste in the water, which will like shielding the bacteria and may influence the UV disinfection effects. So like further study like will need to be done to analyze the real like conditions UV disinfection with the ARB. So that's my work. Thanks for your time to listening.
Thank you.
Hi, I'm Amanda. I'll be short. I was wondering what genes you looked at specifically for qPCR.
So I forgot to introduce here. As I talked about the Bacillus subtilis. So what caused the resistance of this bacteria is that in the genome of this bacteria, there was a like base pair deletion of genes, that is AT deletion of the genes at the BLTR promoter range. So like in order to target the like deletion part, like there are like primer designed to target the deletion part like across the promoter range. Yeah.
Great presentation. Knox, I'm with UNC. So I had two questions. One was the bacillus you were using a vegetative or was it a spore? And then the other question was, why do you think, so if 254 has higher absorption of DNA, why do you think 222 was more effective?
So actually, so first, since this is a vegetary like bacteria, not like cannot have spores for this Bacillus type bacteria. The second question is like if you observe this picture, I only show the like absorbance of DNA like it has a higher absorbance at 254. But if you look at the combination of DNA and protein, so the DNA and protein will have a similar absorbance at 254, but it still has a higher like absorbance for DNA and protein at 222 nanometers. So this means if you look at the compare only the DNA the DNA and DNA protein, the like combination of protein and DNA on like at low wavelengths will have a better performance. So that's our hypothesis, and then we did the experiment to like validate if our hypothesis is right.
Thank you.
Okay, thank you again.
Thank you.
The next presentation is from Valerie. Sorry. Lonnie.
Yeah.
Chid Z. Usama. And he is from Malawi University of Business and Applied Sciences. His title is Individual Risk Perception and Antimicrobial Resistance.
Yeah. All right. Thanks very much. As I said, I am Kondwan Chidziwisano from the Malawi University of Business and Applied Sciences in Malawi. And I'm here to make a presentation on a research about risk perception and the role of environmental factors in the spread of antimicrobial resistance in Malawi. So globally, it is estimated that about 700,000 people die annually due to medicines no longer working on curable infections. And this has been related to antimicrobial resistance, which is an emerging public health problem associated with so many factors, including inappropriate prescription of medicines. And the risk of fecal human exposure via the environment has been reported. And importantly, animal feces has been known to be one of the major contributors to environmental contamination. And poo containing ESBL, which is an indicator of AMR pathogens, has been isolated, more especially in low and middle income countries of the world. And in Malawi, where this study was conducted, if we look at the data collected between 2003 and 2016, there has been high levels of ESBL isolated from patients from a study that was conducted in one of the referral hospitals in the country. It is against this background that a team of researchers from the Global North and Global South conducted a research, the DRUM project, with the purpose of understanding the risk for MR and determinants of exposure, with the purpose of generating data to inform intervention development to reduce the risk of antimicrobial resistance. I should say that the DRUM project was implemented through 5 work packages, and the data I'm sharing here today is it's just part of data through in work package 1. And if you want to know more about the DRUM project, you can follow the link on the slide there. This research was conducted in the southern part of Malawi, in 3 study locations, that is the peri-urban, urban, and the rural locations. And I should say that during the time of the study, sanitation coverage was quite high across all the study locations, over maybe 80%. However, most of the toilets were traditional and improved latrines that are prone to collapse during the rainy season. And we noted that in the urban locations, such toilets were observed along the river for easy emptying, taking advantage of the rain, especially during the rainy season. And such kind of positioning the toilets just increases environmental contamination. An example is the one that we see here. So those are the type of toilets, unimproved traditional toilets positioned along the river, and then the contents are washed into the river, which increases the contamination itself. So the DRUM project collected samples from various sources, as we can see here, for ESBL analysis. And from the findings, I should say that findings indicated high levels of ESBL in animal and human feces, also in the water environment, which included both river and drain water. And we can find out more about this on the link provided there. But related to this, observations indicated that people interacted with the river and drain water, and also animal feces, as we can see from some of these pictures that were taken during the study. Thus, the findings confirm that liver and drain water, including animal feces, provide risk to MR infections among the study participants. So with this, we conducted this research to respond to some of these specific questions. Firstly, what is the level of MR risk perception among the study participants? And also, which are the environmental related psychosocial factors facilitating transmission of MR that is relating to river water, drain water, and animal feces? And importantly, which behavior change techniques should be considered in an MR awareness promotion package? So for us to respond to these questions, we used the runners model of behavior change just to respond to those specific questions. And I should say that we recruited 300 households, 100 from each location. Our plan was to have 600 study participants, 2 from each household, a male and a female, and one of them being head of household and another the caregiver. So the caregiver in this case, we are looking at someone who is responsible for preparing food and also maybe cleanliness of the household. However, we ended up recruiting 529 participants, mostly because the head of households sometimes could not be available, especially in the urban locations, because they also have to go to work. So in terms of the findings, I should say that we noted that slightly above half of the study participants were aware of what an MR is, more especially the females and the caregivers. Generally, most of the caregivers were also females. And the AMR risk perception was low across all the study locations with no difference between male and female. However, the caregivers showed high level of risk to AMR infection than the household heads, and most of the household heads were the male, in this case, were males. We also wanted to assess the source of knowledge about AMR awareness, and we checked if those who ever heard about AMR also knew someone with an AMR infection. And as we can see, we noted that majority of those who ever heard about AMR also knew someone with an AMR infection. And this indicates that the level of awareness about AMR is low, and mostly depends on someone to know those with an AMR infection. So the general public may not have that information about AMR unless they know someone who had an infection. About the perceived source of AMR pathogens within the household-related activities, the participants did not highly perceive the household activities being responsible for AMR infections. Very few perceived animal feces, liver, and drain water as responsible for AMR infection, as we can see from the findings there across all the study locations. They could not really relate these to AMR infection. And for those who mentioned toilets, for example, or those who mentioned drain water, they could not specifically indicate how this contributes to AMR. For instance, if someone is using the toilet, how did this contribute to AMR infection? So what it implies is that they had this information maybe from previous exposure to health messages, maybe about diarrheal disease prevention, but they could not really explain the link between maybe toilet use and transmission of an AMR infection. In terms of psychosocial factors influencing use of river water, drain water, and animal feces, generally similar psychosocial factors were found across all the 3 study locations. As we can see, for instance, this slide is showing findings related to use of river water. Vulnerability was noted in all the questions, implying that those who used river water felt that they were not at risk of contracting AMR through such practices. Similarly, we noted similar findings across all the study locations related to norms, ability, and self-regulation factors. And in the interest of time, I will not be able to show similar findings for drain water and contact with animal feces, but we ran the same kind of analysis, and we identified behavior factors for those 2 other behaviors. So thus, with the identified behavioral factors for each of the behaviors, the learner's model was referred to provide guidance on specific behavior change techniques required. Almost having similar behavior factors for the 3 behaviors in the 3 study locations indicated that behavior change package will not be different. So we may need to have similar kind of behavior change techniques applied in the urban, peri-urban, and rural locations in this case. For instance, here, with the reference of the BCTs in the learner's model, BCT 1, 2, and 3 would be applied to present facts and create awareness about personal risk to AMR infections through specific channels of communication. And in this case, we may refer to group meetings or posters, just to raise the level of awareness among the study participants. And similarly, corresponding BCTs for the norms, attitudes, and the self-regulation factors need to be applied with the use of specific channels of communication if we are to change their behaviors. And why is it important to change their behaviors is because we may want to maybe improve infrastructure, but looking at the economic situation in the country, that may take time. So it is quite important to put much effort as well on issues to do with behavior change so that people should be at low risk of contracting AMR-related infections. And importantly, as we have seen, it will be quite important for such kind of interventions to focus much on men rather than the female counterparts, because men have really shown not to have much knowledge about AMR, and even the risk of getting an AMR infection was quite low among men compared to the female counterparts. So the men should be given a priority to be targeted with these interventions. Maybe a question could be, how do we want to target men? Because in most cases, they are not available for such kind of an intervention. So that's something that people want to think about. Lastly, I would like to thank the Medical Research Council for funding the research, and also recognizing all the important institutions that took part in this research. And if you want maybe to continue engaging, then you can reach me through that email address. Thanks very much for your attention.
Thank you very much. And this paper is open for comments and questions. There's one.
Thank you. That was a super clear presentation, and I appreciate it's kind of early stages. But I'm Esther from UNICEF, as Kondrani well knows. From my perspective, working in kind of innovation and looking at early stage where we should be investing, because I know antimicrobial resistance is going to be the next big problem that my country officers are going to come to me and say, hey, have you got a solution? So based on your early level findings, where would you say the best investment we've got in beyond behavior change as one solution? Where else could we be looking for possible interventions for programs to be ahead of the curve on this one? Thank you. And you can say if you don't know, I appreciate the left field question. Thank you.
Yeah, all right. Thanks very much for that. Indeed, I may not really have a direct answer for that, because what is in my mind is to do with behavior change. And I look at that as maybe a proper solution, because the other alternatives could be improvement of infrastructure, as I've already said, which may require more investment compared to issues of behavior change. Yeah.
Yes.
Great. Thanks very much for your presentation. I'm Saskia Nowitzki. I'm at the University of Oxford, and I've done a little bit of work as well on behavior change in response to water quality risk information from water quality test results.
Yeah.
One of the things I found is the sort of relative empowerment of different households or different people. It has a really big influence on whether they are kind of able, whether they have the self-efficacy to sustain behavior change when information about risk is provided to them. So I'm just wondering if that came up a bit in your use of the RANIS model and with your recommendations of the different communication channels. Does your work is there an element of considering the empowerment of the people that you're communicating the risk information to? So it's a bit of a convoluted question.
Yeah, I think that issue was identified, especially through the behavioral factor, the norms, because under the norms, we had two major issues. We had the descriptive norms and the injunctive norms, whereby those who are in authority, do they approve or disapprove the community members to use, for example, river water or drain water, or maybe do they say something about in contact with animal feces? So it was found that those in authority, they don't really take much responsibility. And in this case, that affects as well the self-efficacy of the individuals. And if that has a role, if we can strengthen that part whereby the local leaders take the responsibility, it may have a role maybe as well to improve the self-efficacy of the individual on how they perceive river water, drain water, and animal feces in relation to AMR infections.
Yeah, really interesting. Thank you.
Yeah, thank you.
Okay, thank you very much.
Yeah.
So this is the time for the next roughly 10 minutes when I would ask the presenters to come up and have a seat. And then we're going to have panel discussion as well as comments and questions from the general audience. So in this session, you see that there are different aspects of AMR that the different presenters have covered, including behavioral risk factors, as well as treatment efficacy factors for particular types of treatment, disinfection of water. And then we have another presentation that has to do with frameworks and policy related aspects. So Um, I'm not sure that any of you have any comments already about the relationships between these things, among these things, because we're looking for global integrated approaches to address AMR. And I would ask, to what extent do you think there are possibilities to look at this so-called One Health framework holistically, given the different perspectives that we brought from the organization? Any comments or questions? Please.
That'd be great.
Yes.
You pass it down. Thank you.
Yeah, I think it will be quite good to look at issues of AMR as a one healthy approach, like what the first presenter talked about. I'm saying this with reference to what I have shared as well, because previously the people were looking or have been looking at AMR maybe as a result of inappropriate uptake of medicines. But we have seen that even issues of wash do play a major role as far as AMR transmission is concerned. So if we are to really win this battle, we may need to look at a number of issues. We need to be holistic in our approaches or our interventions rather than focusing on one issue. Maybe the question could be, because if we're looking at one healthy approach, we are trying to bring in different players into this. Maybe how can we bring all these into one and then move together as a team? Maybe that's where maybe we may have some challenges, but it's quite good to move as a one healthy approach.
Great point. Other comments?
Have anything. Just kind of going off of that, and what a lot of my research kind of focused on is globally, there is that guidance through those global organizations and that tripartite. But a lot of those are focused on just certain sectors. So that holistic approach to AMR, and then also incorporating surveillance, prevention measures, mitigation, and policy. You kind of need a combination of all of those to really combat and manage this global crisis. Kind of the trajectory of where everything's pointing, and we know that, but there needs to be that action still is where we're at.
Yeah, like what I talked mainly focus on the treatment. That's like the antibiotic distribution is like they have different patterns around the world, like even different like in the same like district. So their surveillance like technology will like help us to like have a better understanding on our treatment technology, or like will give us feedbacks of how to do next step to like promote the like treatment or like monitoring of the like ARB and ARG distribution. Thank you.
Great, thank you. Comments and questions from the audience. So we have the speaker's perspective, but some of you may want to say things in response to their perspectives, as well as your own perspectives of the linkage between these three talks and holistically the One Health framework that is being woven with the movement.
Thank you so much for all of your presentations. They were really fantastic. It was great to hear. My name is Nadja Minzermann. I'm working with the GIZ on the sector initiative on One Health there. So I was really interested to see some of these research results. We're looking at specific examples on the ground and how that can be integrated into the global frameworks and how that's going to be led on further in future. I think what you pointed out, sorry, I've forgotten your name, the first presenter, was really important because there has traditionally been a lack of focus on the environmental aspects within the One Health approach, especially in the AMR space. But now, as you surely know, the new quadripartite, the integration of UNEP into the quadripartite or the previous tripartite, I think is a good way to more strongly place the focus on the environmental dimension of these aspects. So maybe, yeah, I think that we really need a little bit more of these hands-on research results to guide the way forward there and to place more of a focus on the importance and the relevance of the environmental sector. So less of a question, more of a comment, and thank you very much for your inputs today.
Hi, thank you guys so much for your presentations. I think for someone who is looking at the agenda that we have for this conference and then seeing where AMR fits in that, I think I had the pleasure of talking to Rachel earlier today and just learning about AMR before I walked into this room. And I don't know if this is the room I would have picked beforehand before I was like 700,000 people, that statistic, and just seeing how large of a problem it is. I'm an environmental engineer, so I do work with environmental health that side, but I also really concentrate on WASH a lot. And I was wondering, how did I miss that or seen that number? And I don't like to stick with statistics and that be something I really said, but that was that's a large number. And so I was wondering, you know, if someone misses this particular section, this side event, right? How would you like to see this type of information integrated into other WASH maybe side events that are occurring right now, or the sanitation or the hand hygiene? Where's the easiest way to integrate this knowledge so it disseminates past this room? Because I think people could really miss, you know, this really important topic if they miss this really specific room where they put all of you guys together, which is great for me to learn, but definitely think could be useful to a lot of people.
Great comment. Thank you. Other comments, questions, reflections?
Yeah, as you kind of noticed just from the few presentations here today, AMR is really complex. So I think a great starting point is taking those proactive measures with outreach and education and training from WASH, because it does tie into it from hand washing, but that also can contribute to AMR from those disinfectants and things like that. So main thing is the lack of education awareness, which you even noted in your study. So I think that's kind of the first step there, but then we also need to prioritize surveillance. So there is a global surveillance program, but we have to understand what's there before we can even start to make policies and regulations. So those are kind of the first two steps that need to be addressed and bring awareness, I think, to everyone about this.
Hello, Connor Lamontagne from UNC. Great talk to everybody. And like you were saying, this really does show like the complex nature of AMR. And so I'm really interested in science communication and the question, because the environmental aspect and the environmental dimension is so ignored, as you showed, a lot of these kind of policy things around the world. So despite the fact that we know that the environment is incredibly important in this, so how would you make that pitch to policymakers? How do you say, this is incredibly important stuff that we need to focus on? Because I think a lot of people are sort of confused by a lot of the sort of lack of information out there and some of the big questions about AMR and environmental parts of AMR. So how would you sort of make that case?
Yeah, thanks very much for that. I think there are a number of ways we can try to raise awareness about environment and WASH or AMR. I should say that one way is to conduct more research in this area that is context specific, to provide more evidence. And then with the findings that we are getting, we may want to engage the policymakers, because the way we have engaged the policymakers maybe on issues of hand hygiene, for example, we may want to integrate issues of AMR together with other WASH interventions. That's the only way, because we don't need to look at issues of AMR separate. It may not work, because that would be very difficult. And it is something that is affecting almost everyone. So if we are doing other interventions in WASH, let's try to incorporate issues of AMR as well, because evidence is already there. In that way, maybe we may scale up awareness of this problem, and then people may take responsibility. Thank you.
And just to add to that too, kind of as I mentioned through my presentation, the environment's overlooked pretty much in everything, not just in AMR when it comes to policy and health. So you basically have to tie it into how it's going to affect people, because that's what we care about. Like, we know it's there, that's great, but how is it impacting me? So tying that together with public health through those WASH measures and education and awareness, I think, is really what needs to happen to make people care about it. At least from my experience, that tends to be how it works.
I think like talk about antibiotics, if you ask some people, they may don't know, they may only know all the antibiotics is effective and is an effective drug, but they may don't know the adverse effects of the antibiotics. And if I know the like US has regulations of the use of antibiotics, but in China, like you even don't need a prescription to buy antibiotics. So I think like we can like let more people know the adverse aspects of the antibiotics and also like regulate the use of antibiotics.
Great, thank you. So just a final comment. First, thanks to all of the speakers and to this highly participatory audience. As I think everyone has mentioned so far, we need to engage everybody globally in addressing this issue. You know, here in the United States, for example, we have public service messages. Things of importance, including COVID-19 and maybe some other health related issues. But we don't have such messages on a consistent and continuous basis for antimicrobial resistance, despite the significance of this as a public health threat that we collectively need to address. And that includes bringing these messages into the classroom, into education of young children and their families. So I think as a final reflection, we could hope that more attention would be paid to connecting what we have here is a fairly high level of discussion among pretty much experts to the communities from which we come and the agencies and organizations that we represent. So thank you all for your participation. I know it's poster time and beverage time, so I don't want to keep you any longer, but thank you very much for coming to the. So one of the other, just one of the other professionals. She's an Asian woman. Yeah. You remember? Yeah.